So we're going to go through the different types of enzyme regulation. The first one called, and I'll go through examples later on, but I'll give you an overview here. First one is called proteolytic cleavage to activate. That means an enzyme is produced as a zymogen, called zymogen. It's produced in an inactive form. The way you activate it is you truncate or clip off a short segment from the amino terminus of that zymogen to convert it into the active enzyme. In fact, most of our proteases in the small intestine are released that way as zymogens. Inactive, once they hit the small intestine lumen, they're activated in situ and they are able to digest the dietary proteins. Okay, very important. We'll give you an example in a little bit. The next kind of enzyme regulation is called covalent modification. And that's also extremely, extremely important. Half of the enzymes we cover are subject to covalent modification. So I want you to understand the term. The reason it's called covalent modification, you are creating a new covalent bond to the enzyme. Typically, that covalent bond, the most common type is the addition of a phosphate. That phosphate typically comes from ATP, and you need an enzyme to add that. So you need a protein kinase. Okay, that's the class of enzymes I mentioned in the beginning of this chapter. Okay, so I want you to think about you're taking an enzyme, you're going to modify the R groups of the amino acids in that enzyme. What are the R groups that can be modified? Let's say that you can add phosphate to. Okay, good, nice. Okay, good. So hydroxyl group, serine can become phosphoserine. Threonine can become threonine or tyrosine. The outcome of that typically can be either to activate an enzyme or, depending on the identity of the enzyme, it can inhibit certain enzymes. So covalent modification is extremely important regulatory mechanism. And the most common one is phosphorylation. There are other forms of covalent modification. That means you add a different group. But phosphorylation is the one of the most important ones. Now, the reverse is true. If an enzyme is in the phosphostate, phosphoserine, and you remove that phosphate, you are breaking a covalent bond. So that's still called covalent modification. Okay? So either way, it's called covalent, whether you're adding a phosphate or removing a phosphate. Okay, third one is pretty easy. Sequestration. Some enzymes maybe exist as a single subunit or multiple enzyme molecules forming a polymer. In some cases, when you form the polymer, the enzyme becomes inactive. So that is called sequestration. You put many enzyme molecules together, so individually these enzymes become inactive. The fourth type here, allosteric regulation, is absolutely very important. Allosteric regulation is where a small molecule, typically again organic molecule, that is distinct, different from the substrate for that enzyme, binds to the enzyme at a site distinct from the binding site, the substrate binding site. It's called other site. And that's the term, what the term allosteric stands for. A small molecule binding to another site on the enzyme besides the enzyme active site, and changing the activity of that enzyme, either positively or negatively, activating or inhibiting. And that's, again, a very important regulatory mechanism. So I really want you to compartmentalize each of these ways. Don't mix them up. There's no one is covalent modifications is one way. Allosteric is another way. Don't mix them up. You want to make sure you know what they mean. So we'll cover some of that, some examples also with allosteric regulation. Now, the last two are kind of related. The last two, I'll start with the first one, and that's called, again, pay attention to the term, induction or upregulation. By definition, what that means is you increase the number of enzyme molecules in the cell. How do you increase the number of enzyme molecules in the cell? By increasing the expression of the gene that encodes that enzyme. That's the only way, that's the only thing that induction and upregulation means. So, for example, let's say the liver, under specific conditions, has 2 molecules of an enzyme called glucokinase. Under another condition, the liver upregulates the gene for glucokinase and makes 5 copies instead of 2 copies of that enzyme. So each cell will have 5 instead of 2. That's what you call upregulation or induction. When you do that, you're not necessarily activating the enzyme, but you're providing the cell with more enzyme. That means there's increase in overall enzyme activity. Okay? So this is very important. Now down here, this is called repression, which is the reverse. That means under certain circumstances, the cells may decide to downregulate the expression of a specific enzyme. We don't need that enzyme, so the cell doesn't produce that enzyme. Okay, or reduces the amount of that enzyme. That's specifically called repression or downregulation. Okay? These last two I want you to pay attention to are the slowest of all enzyme regulatory mechanisms. The reason they are slower, because obviously they involve multiple steps. You have gene expression. That means you have to transcribe the gene. Then the transcribed gene is going to be translated. The messenger RNA is going to be translated into protein. The protein is going to be synthesized in the ER and is going to be modified. So all of those steps take more time. So these last two usually are the slowest, meaning they can take several hours, in some cases days.